I got interested in this project in elephants because it's very difficult to treat bleeding disorders in elephants. I'm a small animal veterinarian, and when we have animals who have bleeding problems, it's usually because of a coagulation issue, so either a problem with platelets in the blood or with coagulation factors. And we have the opportunity to treat those animals with blood transfusions or plasma transfusions to replace the things they need. Something that's a little more difficult to do in wildlife, especially wildlife as large as elephants. We got interested a few years ago in looking at a different part of the coagulation system that we could potentially target in small animals called fibrinolysis. And fibrinolysis is just clot breakdown. So every clot that's formed in the body is ultimately broken down by the fibrinolytic system. There are some drugs that have been used in people for many years to try to inhibit clot breakdown in people who are having bleeding problems. And we thought it would be interesting to look at these drugs, first in small animals and now in elephants, as an alternative to treating with products like plasma and blood that are very expensive and that would require large volumes to treat. And our first step to investigate this was to determine what normal fibrinolysis was in elephants, and also to determine whether drugs that we can use to inhibit fibrinolysis might be useful in elephants, and if so, what concentrations of those drugs would be needed in the plasma in order for it to be effective.
And we were interested in looking at the drug aminocaproic acid to see if it would be effective in preventing bleeding disorders in elephants. Aminocaproic acid is a drug that works to inhibit clot breakdown, or a process called fibrinolysis. So to do that, we used this tool called thromboelastography, and this gives us a global assay of clot formation, clot strength, and then clot breakdown. So these are our TEG machines. They're made by a company called Hemoscope. And I'm just going to sort of walk you through today how the thromboelastograph assay works and how we got the data that we used in the project. So it starts with a sample of elephant plasma, and this is from a group of healthy elephants at the zoo, and we pooled it all together so that we had one big pool of healthy elephant plasma. And then we have to add several reagents to that in order to run the TEG assay. The first thing that we add is tissue factor. So tissue factor is produced naturally in animals in response to damage to blood vessels, whether that's trauma that damages a blood vessel or an infection like bacteria or a virus that damages a blood vessel, and it stimulates clot formation. The next thing that we added for this assay is tissue plasminogen activator, or TPA. TPA is produced in real animals to break down blood clots. So we wanted to, alter our plasma sample so that there was a lot of active fibrinolysis, and that gives us an opportunity to measure the effect of aminocaproic acid. So now we've taken a healthy plasma sample from Asian elephants, we've added tissue factor and tissue plasminogen activator, and now it's more like the plasma sample from a sick elephant, and that'll allow us to measure rapid clot formation and clot breakdown using the TEG machine. So the last step is to load it into the machine. And then we can go to the computer software and hit start. And then that assay runs, and as you'll see on the screen, we can see a graph of the clot forming in the cup. The next step of our project was to take this model of hyperfibrinolysis, or model of a sick elephant's plasma, and see if we could fix it by adding aminocaproic acid. So for that step, we went through the same process of adding tissue factor and tissue plasminogen activator to the plasma. And then we also added aminocaproic acid, and then ran the assay again. And as you can see on the graphs, we get a really different TEG tracing, where you don't have that clot breakdown that you saw in the first tracing.
So in the elephant plasma, we actually found some interesting results that were different from what we found in dogs. We actually found that they broke down their clots a little more easily than dogs did, and so we had to use less of the TPA in our assays for the elephants than we did for our assays in the dogs. So that was our first finding. And we also found interesting results there, mostly that compared to people and to dogs, it took less aminocaproic acid to inhibit the fibrinolysis than it would have in people or dogs. And so I think that was an important finding as well, because if we decided to try to dose the drug based on what we know about people and dogs, we probably would have overdosed the elephants and given them too much of the drug. So I think this was a really useful finding. So it's a great first step in terms of trying to get to the bottom of whether we can use this drug in elephants who are bleeding. I think the next step is going to be to actually do some pharmacokinetic studies where we give the drug to some elephants and we measure plasma concentrations to try to figure out what type of dosing scheme we'll need to use to get them up to the therapeutic concentrations in the plasma we determined in this first round of tests. So still more work to do, but I think really important groundwork to get ready for that next step so that we can start to use these drugs potentially clinically in elephants that are bleeding.
So in conclusion, our thanks go to each of you at the International Elephant Foundation for your trust and your support of our work and your amazing generosity. We hope our contribution will help clinicians and researchers manage better the EEHV disease. Thank you very much from all of us here.